Beyond Species, Functional Diversity and the Maintenance of Ecological Processes and Services by Marc Cadotte et al., published in 2011. Summary. The goal of conservation and restoration activities is to maintain biological diversity and the ecosystem services that this diversity provides. These activities traditionally focus on the measures of species diversity that include only information on the presence and abundance of species. Yet, how diversity influences ecosystem function depends on the traits and niches filled by species. Biological diversity can be quantified in ways that account for functional and phenotypic differences. A number of such measures of functional diversity, FD, have been created, quantifying the distribution of traits in a community or the relative magnitude of species similarities and differences. We review functional diversity measures and why they are intuitively useful for understanding ecological patterns and are important for management. In order for functional diversity to be meaningful and worth measuring, it must be correlated with ecosystem function, and it should provide information above and beyond what species richness or diversity can explain. We review these two propositions, examining whether the strength of the correlation between functional diversity and species richness varies across differing environmental gradients, and whether functional diversity offers greater explanatory power of ecosystem function than species richness. Previous research shows that the relationship between functional diversity and richness is complex and context dependent. Different functional traits can show individual responses to different gradients, meaning that important changes in diversity can occur with minimal change in richness. Further, functional diversity can explain variation in ecosystem function even when richness does not. Synthesis and applications. Functional diversity measures those aspects of diversity that potentially affect community assembly and function. Given this explanatory power, functional diversity can be incorporated into conservation and restoration decision making, especially for those efforts attempting to reconstruct or preserve healthy, functioning ecosystems. Introduction. Throughout the history of biological thought, recognition of the importance of species traits and ecologies informed our understanding of speciation and how species interact with the world. For example, the heretic Roman emperor and naturalist Frederick II rejected the Aristotelian classification scheme based on species essences for one based on physical characters and behavior. In this tradition, community ecologists are increasingly realizing that a trait-based, casual view of community diversity may be more meaningful than species richness or composition. Ecologists routinely lump species together according to similarities in function or in response to abiotic conditions, but the use of traits in ecology has greatly expanded recently. Ecologically, a species is a collection of individuals with phenotypic and behavioral traits that determine when and where they can exist and how they interact with individuals from other species. This view of species as an assemblage of traits is reshaping how ecologists measure diversity, assess coexistence, and restore habitats. Most importantly, though, is the emerging view that by measuring and understanding trait diversity, we can make better conservation and restoration decisions. A trait is any measurable feature of an individual that potentially affects performance or fitness and can be physical, biochemical, behavioral, or temporal or phenological. Depending on the exact nature of the measured traits, traits can influence environmental tolerance and habitat requirements. Thus, traits determine where a species can live, how species interact with one another, informing the strength and excess of competition or the consumption efficiency of a predator, or even the contributions of species to ecosystem function, for example, through their differences in nutrient use and storage. With the increasing awareness that species traits influence coexistence and ecosystem function, ecologists now quantify trait variation or multivariate trait differences within a community, generically referred to as functional diversity. Functional diversity is measured in a multitude of ways. Technically, it represents the diversity of traits, but is taken to represent the diversity of species niches or functions. As a representation of niches or functions, functional diversity has been used to understand how species richness or diversity relates to ecosystem function and how diversity responds to environmental stress or disturbance. The power of functional diversity is that, unlike traditional measures of species richness or diversity, it presupposes a mechanistic link between diversity and the ecological phenomena in question. This intuitive link is attractive, and functional diversity is appearing in the literature with increasing frequency. Figure 1. To advocate for the use of functional diversity measures assumes mechanistic links with niche and functional aspects of species diversity. Here, we review the literature to determine how functional diversity differs from traditional measures of diversity, with a primary focus on richness, given its widespread use, and what functional diversity can tell us about ecosystem function. Are richness and functional diversity correlated? Covariance between species richness and functional richness creates challenges in discerning the dynamics generating patterns and their relative impact on ecosystem processes. The justification for the use of functional diversity measures is a pragmatic representation of potential niche differences, rests on the assumption that variety of functional trait values is likely to give insight into ecosystem function beyond that provided by species diversity measures. Still, several biodiversity ecosystem function studies have manipulated species richness and then deduced functional diversity trends, presuming a positive correlation between these two diversity measures. It is useful then to evaluate the validity of species richness as a proxy for functional diversity. Species richness and functional richness are inherently linked. Monocultures fall into one functional groups, whereas polycultures are usually sampled from a pool of species representing multiple functional traits. Therefore, the correlation between species richness and functional richness theoretically ranges from negligible to a one-to-one relationship. Beyond this, the precise relationship between species richness and functional richness remains unresolved for most natural systems. A study of ant diversity along a successional gradients revealed that the increase in ant species richness and diversity with forest age was mirrored by functional richness and functional diversity measures. Similarly, Hino noted a strong correlation between species richness and functional richness in littoral macroinvertebrate communities. One explanation for a strong positive correlation between functional diversity and richness is a selection effect as defined by Mayfield and colleagues. This selection effect occurs when the likelihood of capturing the range of functional trait values in a regional species pool increases with local species pool size. A random or uniform distribution of species along a functional dissimilarity spectrum would support a proportional relationship between species richness and functional richness. However, this distribution is atypical in natural systems, where species converge into relatively discrete functional strategies. Diaz and Cabino propose a positive linear relationship, with species richness exceeding functional richness. Despite these examples of positive linear relationships between species richness and functional diversity, this trend is not universally supported. Functional redundancy, trait inclusion of measures in functional diversity, the functional diversity metric used, and environmental filters can all shape the relationship between species diversity and functional diversity. Treating species richness as a proxy for functional diversity implies that every added species increases functional diversity by an identical amount. However, in assemblages with functionally redundant species, random species loss is unlikely to decrease functional diversity much, and an influx of species is unlikely to contribute different functional traits to the assemblage, especially when local and regional environmental conditions permit the same values and variety of functional traits. Francesca and Cadotte calculated that 75% of species could be lost from an Argentinian plant community by random extinction before functional group richness would decrease, illustrating the divergence between species richness and functional group richness in functionally redundant systems. If instead species diversity remains static because colonization matches species loss, functional diversity may fluctuate fairly independently of species diversity. This pattern could be seen if functionally unique species colonize an area following loss of functionally redundant species or vice versa. Different functional traits have different levels of redundancy within an assemblage, and there are many possible permutations of the species diversity functional diversity relationship within a single community. When assessing functional diversity, a large number of functional traits increase the ability to detect functional differences between species. That is, it increases the trait space in which they can vary, reducing the impact of species identity and the likelihood of classifying species as functionally redundant. Only traits related to the function of interest should be included in the functional diversity metric. Excessive incorporation of neutral or positively correlated traits will cause artificial convergence of functional diversity and species diversity, while negatively correlated traits will inflate functional diversity values. However, a priori determination of the functional impacts of traits is not likely to be known, but traits used in functional diversity measures should be uncorrelated. The relationship between species diversity and functional diversity is also influenced by the sensitivity of the functional diversity metric to functional differences between species. While Francesca and Cadotte found high redundancy in functional groups, other measures of functional diversity typically reveal less redundancy. Species richness will be greater than measured functional richness when broad functional groups subsume subtle variation between functionally clustered species. In other words, use of functional groups overestimates functional redundancy, distorting the relationship between measured functional diversity and species diversity and probably masking important functional consequences of some traits. When modeling plant diversity patterns, Cadotte et al. Employed plant growth strategy richness as a surrogate for species richness after confirming that functional richness and species richness patterns corresponded. Without this verification, it is difficult to know the actual relationship between species diversity and functional diversity in a given community. Because the species diversity functional diversity relationship is dependent upon community functional redundancy, the number and identity of the functional traits evaluated, the actual functional implications of particular traits, and the functional diversity metric used, Use of species richness as a stand-in for functional richness is ill-advised. The key to understanding the relationship between species diversity and functional diversity and how they affect ecosystem function is determining how abiotic factors influence these diversity measures. The influence of abiotic variables on community composition and species abundance is expected to affect the relationship between diversity and functional diversity. Reported trends between species richness or functional richness and environmental gradients can be confounded by the interaction between abiotic factors and by the span of the gradient examined. Turnover of functional groups along a resource axis may be more informative than total species richness in examining biodiversity patterns along environmental gradients. Surprisingly, few studies have assessed overall functional diversity trends in relation to abiotic factors. Here, we review how nutrient availability, light availability, disturbance, water availability, and temperature affect both species richness and functional diversity. Nutrient availability. Species richness is commonly linked to nutrient availability, but positive, negative, and unimodal trends have all been reported. Klinbecker, Holzel, and Vogel found positive linear correlations between total species richness in peat bogs and availability of nitrogen and ammonium chloride soluble calcium. This trend collapsed when species richness of particular functional groups was investigated. The identity of the dominant functional group varied along gradients of resource availability. In contrast, nitrogen addition reduced species richness in alpine meadows, but alteration to community composition with nutrient supplementation depended upon levels of functional evenness and identity of functional dominance. Functionally diverse communities were prone to composition shifts and increased dominance with nutrient addition because they were more likely to contain a species with functional traits suited to exploitation of the added resource. In fertilization experiments, it is common to find negative relationships between nutrient addition and species richness. High functional diversity in nutrient uptake and utilization strategies could explain the tendency for nutrient-poor soils to foster high species diversity. Quantifying the levels of multiple nutrients may elucidate limiting factors and clarify confounding interactions when exploring the impact of nutrients on diversity patterns. Disturbance. Mayfield et al. Propose a wide variety of relationships between species diversity and functional diversity as communities respond to land use change. Bezos and Malek reported that plant species richness, species diversity, functional richness, and functional diversity were all maximized at intermediate disturbance intensity in temperate upland riparian systems. However, they discovered a disconnect between measures of diversity moving from moderately to greatly disturbed riparian sites. Species diversity and species richness increased while functional diversity and functional richness dropped. Comparing forested and deforested sites, Mayfield et al. Found that functional richness for certain traits increased more rapidly with species addition in forested areas, but this trend was reversed for other functional traits. Disturbance altered the link between species richness and functional richness, and the relationship observed depended on the traits considered. Light, water availability, and temperature. Light limitation can reduce species richness and species diversity. But once again, the impact depends upon functional identity and functional evenness in the community. Light attenuation precludes sibling establishment of photophilic plants. Plants with erect growth forms or that allocate a large proportion of their resources to early shoot growth should be better competitors in low light conditions. As environmental filters such as light act on functional traits, understanding the correspondence between abiotic factors and particular functional traits would help predict which species from a regional pool could colonize and survive an area. Reductions in light, precipitation, or temperature restrict the pool of viable growth habits, lowering species richness. In a natural system in South Wales, Australia, rainforest and eucalyptus tree species richness were both greatest at high temperature and moderate rainfall. However, these two functional types diverge in their tolerance of insolation. Both total species richness and rainforest tree species richness decrease as light levels increase. Eucalypt species richness, in contrast, exhibited a unimodal response. This highlights the importance of examining multiple abiotic gradients in conjunction before making conclusions about biodiversity patterns. Poses and Austin reason that the variety of response curves might be an artifact of the range in resource availability examined. A broader gradient could reveal a unimodal relationship. Peat bog plants of the Kleinbecker, Holzer and Vogel study must cope with stressful environmental conditions, low nutrient levels, and waterlogged soils. The positive linear relationship between species richness and nutrient supply suggests that the entire nutrient gradient studied fell below optimal levels. Species richness increased linearly with water level, indicating that water levels exceeded that which would support a peak species richness. In other words, unimodal trends best describe the changes in diversity along many environmental gradients. Other response curves reported may represent a segment of the encompassing unimodal trend. The above examples illustrate that 1. An assortment of response curves of biodiversity measures to abiotic factors, even within the same abiotic factor, is possible. 2. Abiotic factors interact, and the quantification of multiple abiotic factors should clarify diversity environment patterns. And 3. The different functional types often respond in different ways to environmental gradients, resulting in species richness trends within certain functional groups counter to the overall species richness trend. Logically, an organism's ability to cope with extremes in resource availability should be a reflection of its corresponding functional trait values. How well does functional diversity explain ecosystem function? Since it was first introduced to the field in the 1990s, functional diversity has revolutionized biodiversity ecosystem research. Before they were first empirically tested, theories about the effects of biodiversity on ecosystem function incorporated functional diversity in explanations of causation, recognizing that species functional traits were probable determinants of ecosystem function. When the early experiments were designed, however, they defined biodiversity as species richness, probably for the ease of manipulation. Research was initially motivated by accelerating rates of biodiversity loss that became increasingly apparent over the last several decades. To determine the impacts of these extinctions, researchers created communities containing different numbers of species and compared ecosystem function among these communities. The focus on species richness was thought to reflect real-world biodiversity loss scenarios where species were affected uniformly by extinction. One of the strongest explanations for the biodiversity ecosystem function relationship is that as biodiversity increases, so too does the diversity of functional traits. These traits determine how an organism extracts resources from its environment. Thus, as functional diversity increases, a community increases its partitioning of the total available resources. Griffin et al. Found that the inclusion of highly productive species largely explained the total productivity in macroalgal communities. But for plots that exhibited overyielding, the additional yield of mixtures relative to monocultures, functional diversity provided a better explanation of productivity. In cases where overyielding increases with functional diversity, especially when the assemblage productivity is greater than the most productive monoculture, it is difficult to postulate a better mechanistic explanation than the partitioning of resources along axes represented by the chosen traits. Griffin et al. Cautioned that functional diversity does not necessarily isolate a precise mechanism for overyielding. They were referring, however, to the fact that functional diversity can provide evidence for resource partitioning, but it does not specify along which niche axes resources are being partitioned. A functional diversity productivity relationship invites the conclusion that resource partitioning explains biodiversity ecosystem function relationships, but it does not indicate what the resources are. Nonetheless, when the data for several traits are available, model selection methods can clarify the relative contribution of each trait to overyielding. Functional trait-based approaches can also help identify selection effects. The idea that certain species contribute disproportionately to ecosystem function rests on the premise that those species possess particular functional traits that allow them to capture a greater proportion of the total available resources than other species. Functional traits measurements can reveal whether or not species with similar trait values are contributing the most to ecosystem function. While trait values may elucidate the degree of selection effects in an experiment, multivariate functional diversity measures would not be much use for singling out species because they are community-level metrics. Why functional diversity is important for ecosystem function. Theory predicts that greater differences in resource use lead to increased ecosystem function. The key differences should therefore be in the traits that determine how organisms utilize resources. There are ample theoretical predictions that ecosystem function increases with functional diversity. These predictions are often found in studies that manipulated species diversity and assume that functional diversity was being simultaneously manipulated. In a theory study, Laurier hypothesized that if species have complementary functional traits, such as root geometry, they will occupy non-overlapping spatial niches, and as species diversity increases, so too will the total occupied niche space. In their review, Diaz and Cabido found that one of the best explanations for the effects of functional diversity on ecosystem function was that higher diversity of functional traits increases resource use efficiency in heterogeneous environments. In practice, direct tests of the mechanistic relationship between functional diversity and ecosystem function are more difficult to perform. In one of the earliest such tests, Tillman et al. Planted grassland communities containing varying numbers of functional groups and measured several ecosystem functions. They found that functional richness and composition explained ecosystem function better than species richness. Percy, Hector, and Gaston reanalyzed 6 biodiversity ecosystem function experiments and found that multivariate functional diversity metrics explain variation in ecosystem function better than functional richness or species richness. Functional diversity can explain variation better than richness because it includes a magnitude, figure 2a. For example, if communities in one ecosystem have higher functional diversity than communities in another, that ecosystem should have greater functioning even if functional diversity is strongly correlated with richness in both. If functional diversity lacks variation, that is high redundancy, then biodiversity ecosystem function relationships should be negligible, figure 2b. But if there is variation in functional diversity, it could still explain variation in function even if richness does not, figure 2c. Functional diversity is species blind. Recently, a paradigm shift has begun in biodiversity ecosystem function. Researchers have started including intraspecific variation in their definitions of biodiversity. Driving this shift is an increasing awareness that many of the functional traits known to influence ecosystem function can vary approximately as much within species as they do between species. Furthermore, a large body of literature has emerged showing effects of genetic diversity within species on ecosystem function. These effects are analogous to the effect of species diversity on ecosystem function, and they provide indirect evidence that functional diversity within species can affect ecosystem function. This recent work suggests that it is the diversity of functional traits at all biological scales that is behind the biodiversity ecosystem function relationship. Species richness per se is probably not driving ecosystem function. In particular, Messier, McGill, and Lekowitz explicitly concluded that their detection of substantial intraspecific functional trait variation merited a shift from species-based to trait-based ecology. In at least one study, significant functional trait variation was found within organs of plants. Halshoff and Swenson partitioned the variation in leaf traits across a range of scales of measurement, from organs to species. They found that in certain cases, traits such as specific leaf area could vary more among the leaflets of compound leaves than among species. They also detected significant trait variation among leaves within individual plants and among individuals of the same species. This study demonstrates a lack of evidence for natural breaks in functional trait values at the species level. Functional trait variation is more likely to be distributed continuously along a range of biological levels. In cases where the relevant functional traits are not known or are difficult to measure, species richness may serve as a surrogate for functional diversity. Perhaps more useful than richness, studies linking phylogenetic diversity to ecosystem function also provide indirect evidence that functional traits vary continuously along phylogenies and do not cluster at a particular taxonomic level. It may partly be the ability of phylogenetic diversity to encompass trait variation above the species level that makes it a better predictor of ecosystem functioning than species diversity. The state of the field. Experiments, reviews, and meta-analysis have shown that functional diversity is one of the best predictors of ecosystem function available. Studies are increasingly defining biodiversity as a variety of functional traits in a community or ecosystem rather than a number of species. Underlying this trend is a mounting evidence that taxonomic richness often only weakly influences ecosystem function, particularly at relatively large geographical scales, and universal relationships have mostly eluded ecologists. Evidence for the effect of species richness on ecosystem function is equivocal, and functional diversity is increasingly recognized as a more appropriate metric for biodiversity. In their 2005 consensus of current knowledge on biodiversity ecosystem function, Hooper et al. Proclaimed with certainty that functional characteristics have a strong effect on ecosystem function. In spite of functional diversity's promise, the vast majority of biodiversity ecosystem function studies continue to use species richness. Some researchers have tried to incorporate functional diversity into their experiments using a manually and computationally less intensive version of functional diversity, that is, functional richness. Functional richness provides some of the ease of manipulation of species richness while theoretically capturing more functional variation. Nonetheless, species richness and functional richness are both known to explain less variation in ecosystem function than functional diversity, with functional richness performing the worst out of all measures of biodiversity and no better than when functional groups were assigned at random. Most remarkable, however, is not how poorly traditional diversity metrics perform, but how slow ecologists have been to adopt the more powerful functional diversity metrics. A recent review of 446 biodiversity ecosystem function studies to date revealed that nearly all, that is 94%, focus on either species or functional group richness. For a number of reasons, functional diversity has been met with resistance by ecologists. In some cases, measuring traits may be more difficult than counting species. In other cases, however, measuring a small number of traits may be more efficient than identifying every species in a community. In any case, researchers need to start incorporating functional diversity into biodiversity ecosystem function studies if they are to accurately define the mechanics linking biodiversity and ecosystem function. In the last few years, a promising new tool has emerged for explaining the effects of biodiversity on ecosystem function. The amount of phylogenetic diversity in an ecosystem has proven to be a better predictor of ecosystem function than species richness, functional richness, or functional diversity. Although the specific reasons behind this link have yet to be established, phylogenetic diversity is presumably correlated with ecosystem function because it encompasses most of the functional trait variation in a community, even when it is not known which traits are important, and is therefore a good proxy for functional diversity. Our current understanding of which functional traits are most important for different ecosystem functions and how they should be measured is rudimentary. Consensus is also lacking as to how functional diversity should be calculated. What we usually refer to as functional diversity is an approximation based on a subset of traits of the total functional diversity of a community. Phylogenetic diversity, on the other hand, is a well-established tool that has benefited from a world of molecular methods and data and sophisticated statistical techniques. The two metrics were recently found to provide extremely similar predictive powers for ecosystem function. Ecologists generally believe that phylogenetic diversity is simply a stand-in for functional diversity, while functional diversity methodologies are being refined. In other words, phylogenetic diversity does a better job of approximating true functional diversity than our current functional diversity metrics do. If phylogenetic diversity metrics, which are relatively advanced, are unlikely to greatly improve in the future and functional diversity metrics still require much development, then in the coming years, functional diversity should emerge as a significantly more powerful tool for predicting ecosystem function. Functional diversity in conservation and applied ecology. Conservation and restoration strategies that explicitly account for community functional diversity are ones that prioritize ecosystem function or stability. Increasingly, the goal for restoration projects is to create stable functioning ecosystems. Given that the aspect of ecosystem function that is prioritized can change through time or with human evaluation, and the precise aspects of the biology of species that contribute to ecosystem function are either not known, sufficiently complex, or not generalizable across species, functional diversity offers a methodology to integrate disparate traits and data with limited a priori evaluation or knowledge about the precise functional consequences of particular traits. Functional diversity attempts to quantify overall similarities and differences in species phenotypes or ecologies. Greater functional diversity in managed or restored ecosystems should result in a greater stability through time, as multiple functional traits can help buffer ecosystems against abiotic variation. By quantifying functional diversity in natural communities, researchers gain an additional understanding of the spatial and temporal distribution of biodiversity, which can inform conservation prioritization. Studies examining the effect of extinction on functional diversity reveal that communities generally contain limited redundancy, and that even the random extinction of a few species can result in significant loss of functional diversity. Using high-resolution bird abundance data in France, De Victor et al. Showed that there were significant spatial mismatches in the distribution of functional diversity and species richness. Further, they found that existing reserves seem to protect many of the most spacious sites, but high functional diversity sites were actually underrepresented. Future reserves should attempt to maximize the protection of functional diversity. There is little doubt that ecosystem functions are a top conservation priority because human survival depends on the ecosystem services they provide. These services range from storing carbon to producing food, and there are strong economic incentives to conserve them. Here again, functional diversity offers the ability to understand how diversity relates to these functions. Biodiversity conservation efforts have thus far focused almost exclusively on species. There is evidence, however, that functional diversity is under even greater threat from human activities than is species richness. In a study of the effects of land use intensification on species richness and functional diversity, agricultural development caused significant reductions in both measures of diversity, but functional diversity declined the most. This discrepancy is likely to be caused by the differences we have highlighted in this review. Diaz and Cadotte pointed out that conserving species richness alone will not maintain crucial ecosystem services in the face of continuing global change. They argued that because the contributions of different species to ecosystem function vary so widely, conservation efforts should focus directly on traits and functional diversity rather than on species richness.